All right. Hi, guys. Welcome to our second to last session. This is our 9th session that we've had together. I can't believe it. Thanks for joining. I know we're going to have a good crowd tonight to go over these final FRQs. And then the last time that I see you is on Sunday, obviously the night before your exam. And it'll be a 5-hour session, and there's already 800 people registered. So I'm sure that number is only going to go up. So get pumped. It's going to be crazy. And I know you guys are talking a lot about exam strategies, so definitely keep up with that. The one thing that I can say is that copy paste is definitely the way to go. Yeah, and just keep relying on each other. And I know some of you have already had exams, so congrats on getting those out of the way. Obviously, this is a tough year, and so we're all learning, and you guys should definitely be proud of yourselves. This is it. Last week, well, I guess next week there'll be a few more sessions for exams that are happening next week, but it's our last week, so here we are. Again, 4 to 9 p.m. Eastern on Sunday is our final cram session. More on what that will look like closer to, but should be great. And as always, follow us on all social media. I sent out a recap email yesterday morning around, I don't know, 9:30 a.m. or something like that, with a bunch of different resources, but I used a different internet processor to get to that. So if you didn't get that email, let me know. But you should have gotten an email yesterday morning from me with a recap from Monday night's session. Very similarly to Monday night, we're just doing FRQ strategies and FRQ practice tonight, okay? That's the best thing that we can do. If you're having content problems, go back and watch those content videos, okay? We spent obviously a really good amount of time on Monday talking about standard error. So go back and watch that video if you were confused about that, or do some more practice problems with it. But otherwise, we're just going to keep cruising and keep doing practice, because that's what's really going to be the best for us. Woo. Sorry, guys. You know I have to get my yawn out of the way in the beginning. Okay. So I'm pulling up this slide again. So I know some of you guys were talking about this as well. We have question 1 and question 2. I'm not sure exactly how they're formatting them. It seems like they're having a lot of parts to each question. It doesn't mean that it's worth any more points. It probably just means that they're making each part a point. So I would guess that question 1, is 10 points, which is the same as question 1 in every AP exam. So that's not anything new this year. And it's sometimes only 4 parts, sometimes only 5 parts, just because there's multiple points per part, but it could be upwards of 10 parts if they're just doing 1 point per part. But try not to be too overwhelmed by that, because 10 points is the amount of points that any question 1 on an AP exam would be worth. And then question 2, as I know some of you guys said, I'm so sorry, will probably be 4 or 5 points. And we're going to practice a few questions that look more like what this question 2 might look like tonight. Okay, so everything that I have is tailored to these 2 question types, and interpreting and evaluating experimental results. Obviously, that's what we did all of Monday. So today we'll do a little bit more conceptual analysis. I also have some interpreting and evaluating. I have some experimental design techniques, vocab, that kind of stuff, because some people had asked about that. So we'll go through that at the end. So today, again, we're focusing on this question 2. So they're going to present you with some sort of biological phenomenon with a disruption in it. Okay, so they're actually giving us a lot of information here, and you need to describe the concept, you need to explain it, you need to predict the potential cause or effect of the change, and then support that. So I have a really good example that's very, very similar from years past that looks like this. And you guys should have access to the slides. I realized I didn't press submit on Monday when I was confused why the slides weren't up there, but you guys should have access to the slides underneath, so you can pull these up and be working along with me. And I'll also send you the PDFs from the specific years of FRQs that we're going to be working on. These power words for you, again, we've gone over them a couple times now. But most importantly, we just need to practice, okay? So these first 2 questions are going to be kind of those shorter style questions, okay, that may be worth 4 or 5 points. What I want you guys to do, so make sure that you have paper out next to you. I really want you guys to practice, or pull up a Word document, because copying and pasting is going to be faster. Regardless, I want you to try to actually write out and formulate these responses, because it's almost go time. So it's important that you're getting actual solid practice here. So this is question 4 from 2017. 17. I'm losing it today. And we see this image here, and it says the figure represents the process of expression of gene X in a eukaryotic cell. So part A says the primary transcript in the figure is 15 kilobases long, but the mature mRNA is 7 kilobases in length. Describe the modification that most likely results in the 8 kilobase difference in length of the mature mRNA molecule. Identify in your response the location in the cell where the change occurs. So you could actually just mark this on the cell if this was your AP exam in 2017, but obviously you guys are going to need to name the part of the cell where that's happening. And that's where we're going to start. So describe, remember, you don't need to go into too much detail. You just need to briefly kind of identify what's going on and why we're getting this mature mRNA that's so much shorter. Okay. So that's why I want you guys to start with part A, worth 2 points. Okay. Of 4, we can see 4 bolded words. So this is a 4-point question. And then you can either type it into the chat box or you can write it down somewhere, and we'll review in about a minute. Remember that this is like all tonight is doing this realistic practice. So make sure you're giving yourself the opportunity to have that practice before we go over the answers. Okay, so go ahead and get started, and I will also send you the PDF of this if you want to pull it up on that instead. There's the PDF for you guys. Hopefully you're writing this down somewhere. Whenever you have any thoughts, feel free to write them in the chat box. Is everyone done? I don't see anything in the chat box, but I don't want to go through the answer if you guys aren't done yet. Anyone anywhere? Okay, well, I guess I'll start reviewing it then. I haven't heard anything, but I don't want to make you guys sit here in silence if you're done. Okay, so the first part, remember that describe and identify, if we go back and look at our key power words, they're both very, very short answers. Okay, so this entire question you can answer in like 2 sentences and you can get all 4 points, which is kind of crazy. But for this first part where you have to describe the modification that most likely results, okay, this is the craziest part. You could legitimately just say the removal of introns, and that answer is given to you. If you come up and look at here and look at the color coding, right? Introns are obviously the white and exons are the black, and the mature mRNA is all black. So just by, even if you didn't know what an intron was, you could look at this diagram, okay, and determine, right, that introns are removed. And that would get you a full described point, okay? Again, kind of crazy. So make sure that you're paying attention to whatever diagrams they're giving you, because they're going to give you some sort of diagram to analyze in question 2. Okay. And so you need to feel really, really confident in looking at those and seeing that sometimes the answers might be there for you. So the process, you could say removal of introns, or you could say RNA processing. I know a lot of my students oftentimes mention the spliceosome, and that would certainly count as long as you're explaining what it's doing. So you can't just say the spliceosome, right? But you could say the spliceosome removes introns, or the spliceosome is responsible for RNA processing, or something along those lines. Okay, so that's your describe point. Okay, and then your identification, it happens in the nucleus, right? We know that that's where mRNA is processed. Okay, and then mature mRNA is what will actually leave the cell and be converted into protein. Okay, but that white bubble that we see is the nucleus, and that's where that's happening, because only mature mRNA leaves. So again, you could write, the process is the removal of introns, and it happens in the nucleus, period. So you could actually write just one single sentence and get both points there. So, make sure that you're really thinking and not overwriting, right? I find that I'm always overwriting. Sometimes you just need to take a step back and look at that. Okay, so you should really have more than enough time on this second question, if I had to bet, which is good, because from what I've heard from everyone is that you should actually try to start uploading before the last 5-minute window. Okay, so if you can, try to upload early, from what I've heard. Okay, so then part B, predict. So predictions are going to be a big one. So I think you need to predict in both question 1 and question 2. Okay, so we need to become comfortable with that. The good news is usually any prediction that you make, you can get points for, as long as you back it up. So here we need to predict the length of the mature gene X mRNA if the full length gene is introduced and expressed in prokaryotic cells, and then justify your prediction. So we just practiced this. I think it was last week when we went over this, maybe last Monday. So think about it. What's going to happen if we put this entire gene into a prokaryotic cell? What's the length going to be? Okay. And how do you know that? Okay. So take a minute or two to think about that. Okay. Oh, now I'm seeing all your chats. Holy schmoly. I didn't see any of your chats. I thought I was just talking to myself. This is great. Okay. There was something wrong with the chat. Okay. That's probably what it was. Can someone just type really quick if you guys were able to hear my answer to part A? Okay, perfect. Awesome. I just wanted to make sure that you guys could still hear me. Thank you for responding. I'm glad, because I thought you were just not feeling like answering. And then I looked at the group and I was like, oh my gosh, these are people that I usually talk to all the time. So awesome. Great. Thank you. And I'm seeing a lot of people making predictions that are really, really good for part B. So keep working on that. For that. Good. So Lay has a perfect response there if you take a look at hers, and a couple other people mentioned this as well. Okay, so before we go into the actual right answer, I want you guys to think about this. If they're asking you to predict the length of something in a prokaryotic cell, an alarm should be going off in your head that something's happening here that's different in prokaryotes and eukaryotes, right? Because they're probably not throwing this at you unless there's some sort of difference. I'm so sorry. Oh, what's wrong with me tonight? Unless there's some sort of difference that you should be able to identify. Okay, so that's your first hint. Okay, most people very successfully wrote that the length of the mature mRNA would be 15 kilobases. Okay, Be careful when you say the length of the mature mRNA would be the same, because your reader might not know if you mean the same as the prokaryotic mRNA, which is 8 kilobases, or the same as the pre-mRNA, which is 15 kilobases. So I would say definitely just include specific numbers there, like 15 kilobases, okay? And there's not going to be a poly A tail and a 5' cap on the prokaryotic DNA. That will only be on the mature mRNA of the eukaryotic DNA, but it would not be counted in that initial 15 kilobases. Okay. So that is your length. So your length is 15 kilobases, and your reasoning, and I saw some people write this really, really well, is that mRNA processing does not occur in prokaryotes. Okay, so you can say that there aren't introns or exons. I saw someone write that. That's totally fair. You could say that mRNA processing typically does not occur, or something along those lines where you're saying that that process or that removal of introns does not occur in prokaryotes. Okay, and then you either needed to say that it's 15 kilobases or that it's longer than the mature mRNA in eukaryotes. Either of those would get you the right answer. And Kai is saying she didn't hear the last part of part A. Last part of part A was just that it's in the nucleus, because only mature mRNA that's already processed can leave the nucleus. And the modification is just the removal of introns. And we can actually see that just by looking at the colors in the picture. So just make sure that you're looking at your pictures, because a lot of times they have a lot of key information in them. Okay. Any questions? This, I think, was a really straightforward question, but I'll tell you, and I'll look it up when you guys are working on the next question. I don't think that it scored that highly. So anyway, you guys have an advantage. Dow asks, how can I remember introns get removed? William had a really good way to remember this, and Faith, that's a good way too. I always remember that introns stay in the nucleus and exons exit the nucleus. Introns stay in, exons exit. Prokaryotes do not have introns and exons. They just translate all of their mRNA. They don't do any mRNA processing. And that's a really key difference between prokaryotes and eukaryotes. They stay in the nucleus, Dow, because the rest of mRNA, the purpose of mRNA is to leave the nucleus, to go to the ribosome, right? Or introns go into the trash. That's good too. Yeah, it is the opposite of what you would think, as Ryan said. But introns stay in the nucleus because the mature mRNA is the only thing that leaves the nucleus, because that's where proteins are made. Okay, so I know a couple of you have written some helpful ways there. Something that I want to remind you while we're here, a little content review, since we're talking units 4 through 6, is that an intron is not always an intron, and an exon is not always an exon. So we can make many more proteins. The number of proteins that we can make, let's say, is 100,000 versus the number of genes we have, which may only be 20,000. And that's because we can remove different introns each time. So maybe these parts get removed this time, but next time we might remove different chunks, and that's going to lead to a different mRNA that leads to a different protein, even though we started with the same gene. Okay, so we call that alternative splicing. And it's kind of like how you can edit out different parts of a film and it will give you a different movie, right? You can edit out all the good parts, or you can edit out all the bad parts, and it will make a different movie. That's the same thing here. We have the same starting film, but we're editing out different parts, and it's leading to a different final result. Okay, so just remember that, because that's always an important thing to remember as well. Okay, so do prokaryotes not add the 5' cap or the poly A tail? Angela, they do not. They do not. They have their own kind of way of folding over and adding some things on the end, but it's beyond the scope of this course, so you don't need to worry about it. But they don't have like the typical 5' cap or poly A tail that you think of with eukaryotic DNA or mRNA, sorry. That all happens with eukaryotic RNA processing, okay, which is unique to eukaryotes. Okay. So you don't need to include that in the increasing length. So that's why you can just say that it's 15 kilobases, because that's how long the primary transcript is. So the primary transcript will be the same as what the final transcript is, because we're not removing anything and we're not adding anything. So it just stays exactly the same. Yep. Okay. All right, here's my next practice problem. This one is worth 4 points as well. Okay, so it used to be that questions were either worth 10, 4, or 3 points. This, I'm assuming, again, is like 10 and 5 or something like that. So I'm going to read you through the question. Just as a reminder, hopefully you guys have tuned in before. You know that I skip some words in these questions like this. I'm not going to even attempt this. I'm not even going to sound it out in my brain if this is me taking the APA exam, because it's not worth your time or energy, and it's just going to end up frustrating and confusing you. And then students think that their teachers didn't teach them stuff when really they did. It's just that this is an extension. You're not supposed to know exactly what they're talking about here. Okay, so we'll get there, but I'm going to read through this question with you, and then I'll set you guys to work a little bit. This is kind of like a genetics-y question, and I'll try to find some of the percentages associated with these questions. Okay, gibberellin is the primary plant hormone that promotes stem elongation. GA3 blah, blah, blah is the enzyme that catalyzes the reaction that converts a precursor of gibberellin to the active form of gibberellin. So in my head right now, just so you guys know, I'm writing down on my paper that GA3H converts pre-gib to active gib. That's what I would write down on my paper, right? That this enzyme converts gibberellin to its active form, because I don't want to read that whole sentence again and figure out what this thing does. So that would be something that I'm taking notes on the side, so I can just look and see that interaction when I need to later, okay? A mutation in the GA3H gene results in a shorter plant phenotype. When a pure breeding tall plant is crossed with a pure breeding short plant, all the F1 offspring are tall. When the F1 plants are crossed with each other, 75% of the plants in the F2 generation are tall and 25% of the plants are short. So here I would be doing Punnett squares as well. And make sure, my students would always get confused with like F1 versus F2 versus parental. Okay, so sometimes they wouldn't take it that step further. So we have to do 2 crosses here. We're crossing 2 true breeding plants, and then we're crossing 2 of their offspring, and that's how we're getting the F2 generation. So we're going to need to use that information as well. So these would all be things that I'm writing down on the side as I'm reading this question. And then obviously I see our universal genetic code here. You never have to have that memorized. It will always be provided if you need it. Okay, cool. So first part, the wild type allele encodes a GA3H enzyme with alanine, a nonpolar amino acid, at position 229. The mutant allele encodes a GA3H enzyme with threonine, a polar amino acid, at position 229. Describe the effect of the mutation on the enzyme and provide reasoning to support how this mutation results in a short plant phenotype in homozygous recessive plants. Okay, this actually requires so much thinking. Okay, so I'm going to give you guys a second, because in order to provide reasoning, you need to remind yourself of what this enzyme does. Okay, so this is where those notes would be helpful. And you need to remind yourself what gibberellin does, because that's part of this as well. So you're going to need to go back and do some reading. Okay, this is from 2017, I think. Let me just take a peek and make sure, because I can send you that then, and then you don't have to just look at whatever's on my page. Yes, 2017. Okay, so I'm going to send you this link. And that way you guys can pace through on your own time. Okay. So you're describing the effect of the mutation on the enzyme, and then you're providing reasoning to why this mutation then leads to shorter plants. Okay, reread, reprocess, and then start thinking about what you might include in your response. This is a really realistic practice problem, so definitely take this one seriously. My dog wants to say hi and wish you guys good luck. Here, come here, Addie. She's a really big dog, but she can also be a lap dog. So she says, hi, good luck. She says, I don't want to be on your lap right now. Okay, sorry to distract you. Ooh, Ida. Such a good response. Look at her go. Yes. Hopefully everyone else is writing things out as well. Remember, that's the most realistic practice you can give yourself. She's in here. Yes, Arjun, it was a good answer. Good. Sierra, that's a great answer for providing reasoning. Okay. Very nice. I'll wait for a few more and then we can talk about how to create a really strong response here. So Dow says he's having trouble processing. So Dow, what I want you to think then is, what does an enzyme do? What is an enzyme? And how might an amino acid substitution change an enzyme? So don't think so much about like all of the logistics with the nonpolar and polar and which amino acid it is and all that good stuff. Just think about how a mutation might affect an enzyme. Okay. Okay, good. I'm seeing a lot of really good responses here. So going off of Dow's point, sometimes you read a question and you're just really, really stuck. So that's where I want you to unpack and go back to basic biology, which is what I've heard from readers everywhere, is when all else fails, go back to basic biology. You guys know what an enzyme is. You know that an enzyme is a protein, and you know that the protein is made up of specific amino acids. And then if we think a little bit more complex, those amino acids allow the enzyme to have a very specific three-dimensional function. And the three-dimensional function is what allows an enzyme to do what it does, right? Because it can grasp things in a very particular manner. So if we're changing one of those amino acids, especially if we're changing its chemical composition from nonpolar to polar, we can assume that this enzyme will not have the same shape or the same structure or the same function, okay? And that's actually all you needed to say. Yes, so Kai, the mutation would make it unable to be active, would make it unable to bind to the same substrate. So on the actual scoring guideline, the only thing it has for description is that the amino acid substitution changes the shape or the structure or the function of the protein. So you guys actually went above and beyond in your responses, which is great, okay? But you don't have to go to that level necessarily. So if you just stick with your basic biology, you can still get that point by knowing that a mutation is going to alter the shape of an enzyme, okay? So put that in your brain. It may show up again. They love talking about enzymes, right? And so that's an important thing to keep in mind. To provide reasoning to support how this mutation results in short plant phenotypes. Okay, so a lot of you guys had really good answers for this as well. All in all, you guys did a great job. So this is where I would go back to my diagram and realize that GA3H converts an inactive gibberellin to active gibberellin, which promotes stem elongation. So if we have an inactive enzyme, we're either inhibiting gibberellin or we don't have enough gibberellin, okay? And because gibberellin is responsible for stem elongation, then we don't have stem elongation. And that's really all you needed to say. So in their answer key, they have the mutation decreases or eliminates gibberellin production, and that's it. And sometimes you look at these rubrics and you're like, why is the answer so simple and the problem is so confusing? And that's why I'm just instilling, like, take notes. What does this thing do, so that you don't have to go back and be really, really stressed? You can just say, oh, if we inhibit this, then we're inhibiting the production of this, which leads to stem elongation. And that just makes a lot more sense out of a complicated issue. Okay, same with the enzyme. You guys know how proteins work and their 3D structures. And if you're a little confused on that, go back and watch the Unit 1 review, okay? Because we talk a lot about enzymes. Actually, I think it's Unit 2 now that I think about it, because we talk a lot about enzymes with cellular energetics, okay? So seems super complicated. Answer's not. super complicated. I know. I always overthink them too, Maddie. So I feel that you guys have much more thorough answers than are even necessary, but they're all really, really solid. Ryan, since the grading system is a bit different this year, can we still get the full points by just hitting the points they're looking for? Yeah, so you can always get the full points just by hitting the points that they're looking for. Usually they say less is more, right? Because kids will write like 4 pages for a 10-point question and end up getting like 2 points. So usually they say less is more, but that's really the majority of it. So the rubric itself hasn't changed. So they're still going to be looking for specific points, but you may have different questions in order to get those points. But you're still going to be having those same power words and having usually to get 1 point per power word. So that hasn't really changed. You'll just have a lot more variations of questions and less points total, right? So they'll have to just kind of alter how you're getting your 1s, 2s, 3s, 4s, and 5s, but most importantly 5s. Okay. All right, cool. I'm seeing a lot of really good responses over here also. Thank you for your participation. I appreciate it. Hopefully you're finding it helpful as well. Okay, part B, using the codon chart provided, predict the change in codon sequence that resulted in the substitution of alanine for threonine at amino acid position 229. I like this point. So this is another prediction point. We're going to keep practicing with predictions because it's going to show up twice. Okay, so you need to look at the codon chart and predict what the most likely substitution would have been in order to convert alanine to threonine. Okay, so alanine to threonine, I'm going to switch back, but as you can write it down, ALA to THR. I'm going to switch back to the codon chart for us. Take a second. Take a look. So I'm doing this with you guys just to practice. And right now I'm going and I'm finding alanine and I'm writing down all the possible combinations. And then I'm going to find threonine and I'm writing down, and oh my goodness, look at that. Can we see how easy it would be to get a mutation between threonine and alanine? I can. Yes, good. So remember, we're going alanine to threonine, okay? And so with that, we need to say we're going from a G to an A in the first position, okay? G to A, not A to G. So make sure you're being really careful there. Yes, good. So you can just say the first position of the codon, it's a substitution mutation, but you do want to be more specific and say exactly what base was most likely substituted. And just to give you guys, this is where we're looking, right? And you'll notice that alanine is all C, U, C, A, and G with G's in the front. It's all GC, and then the third base changes, and this is all AC and the third base changes. So most likely the A got converted to a G, or sorry, the G got converted to an A. I just did it too, as alanine got converted to threonine. Okay, so that's what you needed to include. You can give a specific example, Ida, like GCU to ACU, and they do have that as a possible answer, but I think it's easier just to say that there was most likely a change from a G to an A in the first position of the codon. Yep, but you can give a specific example if you would prefer. Okay, so you can say most likely G was substituted with A. For example, GCU may have been converted to ACU. If that makes you feel better, you can certainly include that if you want that little bit more detail. I see another question. Grace says, someone told me that everyone taking the test is going to get a different question. Not everyone, but I don't know how many different questions there are, maybe like 20. I don't know. I know that there were, teachers had entire classes that all had different questions, but it's not everyone. So I know there are a lot of different versions, but I don't know how many. Okay, someone said for Euro, apparently 3. I think Calc was the one that had more, but I'm not positive. Okay, cool. So that would be yours for that. So look for those patterns. And it sounds like some people haven't learned about this in class. So there is a video. I will make sure that I send out a mutations video. Oh my gosh, none of you guys? in my recap. Okay. The good news, though, is here that you can just use the chart and look for differences. I know that's not like perfect, but even if you haven't learned about it, you can tell that the G versus the A is what's changing. But I'll make sure I send a video on mutations in my recap so that you guys can take a look at that, because I definitely want to make sure that you have all these resources for sure. Okay, cool. All right, I like that prediction. I think that's a great point. And then part C, describe how individuals with one heterozygous or two homozygous copies of the wild type allele can have the same phenotype. So basically they're saying, how can a heterozygote and a homozygote dominant both be tall, right? How is that possible? Okay, so this is a huge point that most students missed, and it's something that I always try to cover in class. So I want you to think about it. Think about what a gene does for an organism, okay? There's a very specific thing that a gene will do, okay? When you're thinking about, maybe you've talked about this specifically in class, it's definitely an important concept, okay? But why can a heterozygote and a homozygous dominant individual both have the same phenotype, okay? Think about it for a bit, and then let's write about it. Okay, I'm seeing some answers. I'll give you a little bit longer. I'll give you a hint. It's more than just the fact that it's dominant. What does that mean if an allele is dominant? What does that mean? Addie. Maybe my chat froze again, but I only see two things here. So I'll give you like another 30 seconds just because I'm not sure if the chat froze or if you guys are still working. Totally fine either way. I just don't want to waste your time because we have one more question that's important to go over. Okay, I'm going to jump in because maybe the chat froze again. So I gave you the hint that it's not just that something is dominant or something is recessive, right? Because we need to know what that actually means. Because remember that we're talking about little molecules here that are making protein that are doing things, right? It's not just like one piece of DNA is stronger than another piece of DNA, right? It all has to do with the protein that they make, okay? So usually what happens, to put this into context so that you can use this in future situations or future questions, is that the homozygous dominant allele produces a protein, right? And then the homozygous recessive allele produces a mutated protein. So that's like this. We have this wild type allele that encodes for this enzyme, this active enzyme, and then the homozygous, the recessive allele codes for a mutated protein, okay? So if our dominant allele makes a good protein and our recessive allele makes a mutated protein, okay, when that comes together, right, like with a heterozygote, the dominant allele is able to make enough protein that we don't see a phenotypic difference, okay? Oh my gosh, the chat came back. Wow. See, it happened again. Okay, Cool. Awesome. I'm seeing some really good responses here. And you know what? A guess is okay, right? We're just trying here. But what you really need to write here is about the protein, because the DNA does not overpower. The DNA is not stronger. None of that means anything. DNA doesn't do anything in the cell. The only thing DNA does is code for protein. And the protein is what does stuff. Okay, so the protein either is a good enzyme or isn't a good enzyme. And if it is a good enzyme and you have at least one version of that gene making that good enzyme, that's enough. Okay, so the answers here that would get you points are saying that enough active enzyme is produced from one wild type or one dominant allele, or saying enough gibberellin is produced with one wild type or one dominant allele. Okay. But those are the only answers they accepted because you really do need to take it to that level. So notice that neither of them mentioned DNA. Stop. Okay, neither of them mentioned DNA because DNA doesn't actually do anything here. It's all about the enzyme that's produced, and enough of it is produced in the heterozygous individual that they're still able to have the dominant phenotype because enough gibberellin is produced. Any questions about that? So brown versus blue, it's the amount of melanin in your eye. So it's just another protein that's producing a pigment. So everything has to do with a protein that's a pigment because it's not actually DNA that's coloring your eye, right? It's a pigment that's produced by a protein. So it's just whatever pigment is being produced by the protein that's coded for by your DNA. Okay? Awesome. Sorry that the chat froze up there again. I don't know what's going on. I think Crowdcast has been like super overwhelmed, especially since there's some final cram sessions happening. Okay. Someone's asking, what would you say are the most common FRQ topics that we should make sure we should study? I don't know. It's hard to say. I mean, you need to know experimental design, but I couldn't tell. I mean, you all may have a different experimental design on a different unit. So I can't really tell you a specific topic. You need to be really, really strong in experiments. And then I would say, I don't know, I guess know a lot about DNA and genetics. And it's hard for me to just give you one thing to study just because I have no idea because these are all application based. So they're going to do the same kind of application, but apply it to different situations, if that makes sense. So it's more important that you understand the concepts generally than you need to know anything super specific. Ida asked, do you think it's better to review content or FRQ writing at this point? It depends on how you feel with your content. As long as you feel okay with content, I would say FRQ writing just because last minute you can look up information to fill in the gaps. But if you really don't feel good with content, then I would say start there first, I guess. Yeah. Okay, cool. So we're probably only going to get to do part of this last question, but this last question is really important because it's an experimental design question. And I know some people had asked me to go through some more like specific vocab with experimental design. So we're going to apply this and look at how this experiment was run and provide evidence and factors and all sorts of fun things. We won't have to do all of this. So we'll probably just do parts B, C, and D. So that should give us enough time to finish. But before we do, I just want to go through some vocab for experiments again, because people were asking about this. So you absolutely must be super familiar with this and be able to identify it in any type of experiment. So if you need to practice, you need to practice, but you need to know the difference between an independent variable and a dependent variable. And sometimes my students get them confused, and you absolutely cannot get them confused. So make sure that you know them by Monday. An independent variable, I always taught when I taught freshmen, I always used to say an independent variable is what I change. The IV is what I change, because it's usually what the scientist is manipulating. More generally, it's what affects the dependent variable. Because sometimes when I say what I change, independent variable is often time, because over time, our dependent variable is changing. So we're not changing time, but it's what is causing the effect on the dependent variable. The dependent variable is what we're measuring. So what are we looking at? So if we're looking at the change in plant growth over time, the growth of the plants is the dependent variable and time is the independent variable. If we're looking at how sugar water affects plant growth, sugar water would be the independent variable and plant growth would be the dependent variable because that's what we're measuring and it's what the sugar water is affecting, okay? So the IV affects the DV. The DV is affected by the IV. Okay. Then there are the controls, your control variables. These variables are so important and they're kept the same across all groups. So it's not the same as a control group, it's a controlled variable. So an example from my plant example would be like the same pot, the same type of plant, right? It wouldn't be fair if you were using all different types of plants and then seeing how they were growing, because all different plants grow in different ways. So the same species of plant, the same amount of water, maybe the same location, right? You're not going to have one in the sun and one in the shade. Those are all controls. And the purpose of the controlled variable is to make sure that the only thing affecting the dependent variable is the independent variable. And scientists like ride or die by this, and they have to prove this to the millionth degree, oftentimes in their research, to prove that they have enough controls in their experiment that the thing that was actually doing the changing was the independent variable and the dependent variable. Okay. Okay, so next there's the control group and the experimental groups. The control group is the group that you compare to, okay? So you want to see if the experimental groups that get your treatment are different than this control group, and it usually receives no treatment. There's no independent variable. So if we're talking about like sugar water, maybe the control group only gets water, okay? And then the next group gets some sugar in their water, and then the next group gets a medium amount of sugar in their water, and then the last group gets a ton of sugar in their water. But you're comparing that to the control, which is just a regular plant with regular water, okay? You still need to water the plant, otherwise it'll die, okay? But that would be your control. The experimental group or groups are the ones that receive the independent variable, usually in varying degrees. Okay, and then the last thing is the null hypothesis, which we practiced on Monday. But the null hypothesis is always going to be that the independent variable, whatever that is, does not affect the dependent variable. So in my example, it would be that sugar water does not affect plant growth. On Monday, we did the bee example and we said caffeine does not affect the memory of bees. Okay, so that would be our null hypothesis. But this is like a quick cheat sheet in case you get confused with variables, because I have a feeling you're definitely going to need to name some. Okay, cool. All right, so in this example, obviously we have a lot of data, and it's students studying two different aquatic plant-eating unicellular protist species, A and B. So we know that these are similar species. They're kind of naming some controls. They designed an experiment to investigate the ecological relationship between the two species. Okay, so group one is species A and B grown in separate containers, and group two is species A and B grown together in the same container. And then you can see the amount of individuals over time. Okay, in treatment group one, the student placed 10 individuals of species A into a container with liquid growth medium and 10 individuals of species B into a separate container with an equal amount of the same liquid growth medium. That is a controlled variable. Equal amount of the same liquid growth medium. We're controlling what they're eating. And we have the same amount of species in each. In treatment group two, the students placed 5 individuals of each species into a single container with the liquid growth medium. The students then manipulated the containers under the same environmental conditions and recorded the number of individuals in each population at various time points. The results are shown in table 2, and we can see here that we have a lot more growth from species A than species B, but we have an increase in both over time when they're grown separately. And when they're grown together, we only really have growth of species A. Species B starts to grow and then tapers out, and by 70 hours, there is no more species B, okay? And remember, this is where these two species are grown together, okay? All right. Sorry, yes, this is 2019. I apologize. I should have sent this to you. Thank you to whoever said that. This is 2019 question 2. There's your PDF. Okay. Okay. So the first question is asking you to complete an appropriately labeled graph. And obviously we're not going to practice with that because you do not need to make your own graph. You just will need to be able to interpret graphs and tables. Okay. So we're going to skip to part B. As shown in the table, the student established treatment group 2 with 5 individuals of each species. Provide reasoning for the reduced initial population sizes. Okay, so you need to go back and read, take a look at the experimental design. Why are we now going to 5 individuals of each species instead of 10? And tell me why. Can be a pretty short answer, just 1 point for this. Go ahead and get working on that. Then we'll come back briefly, do parts C and D, and call it a night. While you guys are working, I'm just going to answer these questions really quick. Maddie, you don't need to know the difference between positive and negative controls because all of the controls that you're going to be worried about are negative controls. Okay, so negative controls are what you guys are working with. Grace said, "Is our test definitely going to have an experiment-based question?" Yes, your larger, your question 1, your 65% question is an experiment-based question. And if you go back to one of the first slides in the PowerPoint, it tells you exactly what question 1 is. Okay, good. I'm seeing some really good answers there. Way to go back and read carefully, right? Awesome. We'll go over the right answer in 30 seconds. Okay, so you get your points here for mentioning that we have the same total number of species in every container. We have 10 species in each container to start, right? Because remember in group 1, we have a container of 10 species A and a container of 10 species B. And then in group 2, we have 2 containers of 10 or 5A and 5B. So that's 10, and 5A and 5B. So that's 10. So that's a control that we only have 10 species per container to start. So that's what you needed to say. And I see that most people said that. So 5 and 5 is equal to the total number of the single species in group 1. Okay, so that's a perfect example of where they're asking you to provide reasoning. And the reasoning is that it's a control variable, right? That we need the same starting numbers. Okay, awesome. All right, part C, the student claims that species A and B compete for the same food source. Provide 2 pieces of evidence from the data that support the student's claim. Evidence here means numbers. Anytime you're asked to give evidence, you should provide numbers from a data table or graph, okay? So you should always, always, always use numbers to back it up. So go back and look at your numbers and tell me why we think, what evidence is there that species A and B compete for the same food source, okay? 2 pieces of evidence, so that's 2 points. Okay, I'm bringing it back to this. Remember to provide numbers here. Two specific data points that you want to reference. Okay, good. It seems like most people are getting the reason that they know that A and B are competing because in group 2, species B decreases down to 0 when it is mixed with species A, okay? So that's one specific data point, and you could reference that we start with the same amount of both species, and then we end up with 0 species B and 1,400 species A. That's a really good data point that I think most people wrote about, which is awesome. But we need 2 pieces of evidence, so we need to reference something else as well. Okay, so something else that you could reference could be as simple as, you could also talk about species B, but you could say when species B is grown alone, it can grow up to a rate or up to 700 in that container versus the 0 that are found when it's with species A. Okay, so you can compare species B from group 1 with species B from group 2 as your second data point. So you compared species A from group 2 with species B from group 2, and then you compared species B from group 1 with species B from group 2, if that makes sense, right? So that would be another metric. You could also say that the growth for species A is slower when it's growing with species B than when it's growing alone. Because if you look, and it would be easier if you had already graphed it, but if you look at the growth of species A in group 1, it's much more substantial than the growth we see for species A in group 2. Okay, so you can really compare any of these different columns and get your data points. Okay. Yes, so the groups grown separately would be the control groups in this situation, because we're just trying to see how they normally grow versus when they're grown together, which would be the independent variable. Can we use as evidence that they say in the question that both species are plant eating? No, that wouldn't be substantial enough, just because we don't know what type of plants. There are so many different plants that many things don't compete over. So you really need to use specifics from what we're seeing from the results. Okay, so you always want to look at exactly what's in the results and use that. Okay, good question though. I'm glad you asked. Quickly before we jump into the final part, which will be part D, Lauren asked, should we print out the equation sheet, or do you think that won't be necessary? Lauren, I'm assuming they will give you the equation sheet somewhere. I don't know if someone else can answer who took like calc or something else that has an equation sheet, but if knowing me, I would print it out just because I like to have things in paper better so that I can write on them and just to be safe. Okay, but maybe someone who's taken another exam that would require an equation sheet can answer that question. Oh, it says they don't give you an equation sheet, so you should print it out, because I guess you could just look it up if you needed to. So you could look it up if you needed to, but I would print it out, is what it's sounding like. Okay, Calc didn't give one. That's so rude. All righty. All right, so last thing, D, predict 2 factors that will most likely limit the population growth of species A in treatment group 1. Okay. Ooh, you know what? I'm actually going to skip that for part E. Sorry, because part D, that's more ecology, and it's just like habitat, food, oxygen, like the normal things, and waste. Part E, though, let's do that as our last one. Many protists contain an organelle called a contractile vacuole that pumps water out of the cell. The student repeated the experiment using a growth medium with a lower solute concentration. Predict how the activity of the contractile vacuole will change and justify. This is a great review of Unit 2. Okay, so you don't need any of the information on the previous page. Oftentimes, part E of these long questions will just ask you to connect to a different unit. So you don't need any of that information. You just need to know that our growth medium now has a lower solute concentration. So think about what that means as far as osmotics. You guys know this. How is the activity of that contractile vacuole going to change? Vacuole pumps water out, okay, and justify, okay? Take about a minute. This should be easy at this point because you guys are experts of osmosis. Good. Ida has a great response. Maddie has a great response. Oh my gosh, you guys are crushing it. Great work. Yes. Awesome. So basically everyone is saying that this is a hypotonic environment, so the contractile vacuole will increase, okay? So the 1 point comes from predicting that the contractile vacuole activity will increase, okay? So that's a point. You have a 50-50 chance there because you could write, it'll decrease and then you don't get the point. But just guess something if you don't know, okay? And then to justify, you need to say why you know that. So we know that because we're in a hypotonic environment, okay? But make sure you say hypotonic environment because technically the cell is hypertonic in comparison to the environment. The environment is hypotonic in comparison to the cell, okay? So just to be specific there, we're used to talking about the environment. So say that that's a hypotonic environment or that water would enter the cell. You could also talk about water potential, but I'm sure that's not on the top of everyone's mind. So you do need to mention hypotonic, yes. Okay. Oh no, I see that Derek and Brandon just joined. Sorry guys. Well, hopefully you can join on Sunday. But yes, so you need to mention that it's hypotonic, or you can mention water potential or some other technical one. Water potential, Ida, is just the movement of water. It's always going to move from a higher pressure to a lower pressure, or a higher potential to a lower potential. Okay, so water will move in that direction. And it's just a calculation that's based on usually the solute concentration. So it's just a mathematical way to basically prove like a hypotonic versus hypertonic environment. It just puts numbers behind it. Okay, so hopefully that helps. Why are the cells haploid after meiosis 1, Derek says? Oh, that's really confusing. It's because the homologous chromosomes are separated, not the sister chromatids. Okay, so you have two versions. Oh, you have two copies of one version of that chromosome. Derek, I'm going to email you individually and answer that for you because it's super confusing to understand. So I'm going to email you. Okay. Or I'll answer it at the end. Yes. Okay, cool. So the cell is in a hypotonic environment, okay, which means that we're going to have to be pumping more water out because that's what the contractile vacuole does. As water comes in, we're going to need to pump it out, and water will come in because it's a hypotonic environment. So hopefully you guys enjoyed that question. I do too. Ida says, how many math questions do you think we will have on the test? Maximum 1, right? For sure. Do we think we'll have to do a chi-square, Grace says? I'm not sure. I've been predicting chi-square for the past 2 years and it hasn't showed up. I kind of doubt it that they'll throw that on there just because you have such limited time, especially if they're doing a bunch of different versions of a question. So I say it's pretty doubtful, but yeah. Hopefully that answers. Oh, yeah. Okay. Maybe I'll just, I can try to answer it quickly, but I can just send out an email to all of you guys. So basically, okay, I wonder if I can find a diagram really quick. All right, hold please. If you guys want to hop off, if you're done, because I'm going to be switching topics really quick, feel free. I will not be offended. Otherwise, I'm going to open a virtual whiteboard really quick and try to explain this. Okay. All right, y'all. So I used to make all of my kids, they hated it, but they also loved it. I used to make them all draw pictures of a cell. So I'm going to draw you a little picture of a cell right now. Okay. Hopefully you like it. All right. Hopefully you guys can, I'm going to have to make this bigger. Okay. Wow. Oh my God. My head is so big there. I'm so sorry. All right, here we go. It's because I'm leaning in trying to write on this thing. Okay. All right. So sometimes something that students get confused of often is actually our chromosomes, because you learn that we have 46 chromosomes, and that's true, but it's kind of an oversimplification, right? Someone wants to explain that science, you just get lied to less and less each year, and I find that to be pretty true. Okay, so I'm going to draw you a little cell really quick, and this cell has 4 chromosomes, okay? So we say it has 4 chromosomes, but really it has 2 versions of chromosome 1, one from its mom and one from its dad, right? And this is why we might be like heterozygous or homozygous recessive or homozygous dominant, right? And then we have 2 versions of chromosome 2, one from mom and one from dad. And they have the same genes on it, but they might have different versions because, again, we might be, I'll use different letters. Sorry, I'll use like D. Homozygous dominant, homozygous recessive, sorry, or heterozygous, okay? Okay, so that's the first thing that you need to understand. And these guys are called homologous chromosomes. Okay. So when we first replicate DNA, okay, we boom, get that X formation, okay, because that doesn't happen until we actually replicate the DNA. It's not how our chromosomes normally are, okay? So right now we have double the DNA, okay? Double the DNA that we normally have, and these are our homologous chromosomes, okay? And these are what's lining up at, this is how meiosis is different than mitosis, because this is what's lining up together at the metaphase plate, and this is where we see crossing over occur. But I'm not going to show that right now because, let me just move this guy. Oops, I thought I could move him, but apparently not. I'm just going to move him over. Yeah, there we go. Okay. I'm not going to show crossing over right now for the sake of time. But what happens is in meiosis 1, these guys line up, okay? Boom. Okay. And they're going to get pulled apart. So now and obviously the same thing's happening over here too, but for the sake of time, we're just going to focus on this first chromosome. So now we have this cell that's getting ready to divide and this cell that's getting ready to divide. Okay. So what you guys are thinking is that this is still a diploid cell because we still have double the amount of DNA, but the problem is we only have one version of each chromosome, right? And, technically there's crossing over, and as I said, we're not getting into that, but in these cells that are going to divide, right, we only have one version of chromosome 1 here, okay? So we don't have the diversity. We don't have, we're technically, we only have maybe, big A, one allele for this that's going to eventually get separated. And over here, we only have little a, right? We don't have those two different versions anymore from mom and dad. We don't have that genetic diversity because our homologous chromosomes have separated and we only have one version of chromosome 1 left. So it's the one that's majority from mom or the one that's from majority from dad. And those are obviously eventually going to get split up as well in meiosis 2. And we're going to have, this at the end. And obviously this would be paired with blue and purple. And I have my kids all get, colored pencils out and we do a whole big thing and you can see independent assortment and everything. And I'm not going to go through that all right now. Maybe I'll do that on Sunday if you guys are interested. But hopefully this explains why this, even though we technically have double the amount of DNA, is not technically diploid because we only have one version of each chromosome, and that's what we care about, right? Haploid means we have one version of each chromosome, and diploid means we have two versions of each chromosome. Does that explain your answer? Hopefully it does. I can draw out something. I definitely did go over more of inheritance stuff a couple weeks ago, and I can send that out. But hopefully this kind of makes more sense. So it's really just has to do with the number of versions of each chromosome you have. So a haploid cell has one version of each chromosome. And when we split those homologous chromosomes, because we're not splitting them, so why aren't mitosis cells haploid? Okay, because this part does not, homologous chromosomes don't line up in mitosis. So in mitosis, if this cell was going through mitosis, right, homologous chromosomes do not line up. Each chromosome just lines up at the metaphase plate, like so. We don't do this. This does not happen. Homologous chromosomes do not pair up. Okay, that is something that is solely in meiosis. That's why we have those two divisions. So these guys will get pulled apart. Okay, and when they do, we have one version. We have two versions of each chromosome. Sorry, we have the red and the purple, and we have the green and the blue in the daughter cell. So the daughter cell will have the exact same DNA in it as the parent cell. We have two versions of chromosome 1 and we have two versions of chromosome 2. Okay. Does that make sense, Derek? Cells are the only cells that are haploid are egg and sperm cells. Those are the only cells that are haploid. Meiosis only occurs for egg and sperm cells. No other cells go through meiosis. Haploid, so the only haploid cells are egg and sperm. So they duplicate to make the X in step 1. Yes. Okay. So maybe I need to do a meiosis review on Sunday. I may be streaming the whole 5 hours on Sunday. Stay tuned. I got to talk to Amanda, which would be crazy. But Derek, I think I might just need to redraw a diagram. I forget, I actually may have drawn a diagram out. I'm trying to think if I did in a previous stream. I'll look for what I have. And if I don't have something good, I will make something good or talk about it on Sunday. But hopefully that helps a little bit. Okay. So again, oh, you guys are so sweet. Right. Homologous chromosomes are lining up in the middle for meiosis, and homologous chromosomes do not line up in the middle for mitosis. Okay. All right. Hopefully that makes a little bit of sense. Okay. If you have more questions, shoot me an email. Otherwise, I will see you guys on Sunday for sure. And we'll definitely do more FRQs. Okay. Cool. You guys are great. Have a great night.